Genetics is all about the fundamental principles of inheritance, including what they are and how they come about. It is the science of potential, the transfer of biological information from cell to cell, parent to offspring, and generation to generation. Genetics is concerned with why and how biological information transfers, because biological information transfer is the basis for differences and similarities recognized in groups of living organisms. Genetics explains the physical and chemical nature of biological information and asks, what are the sources of genetic information, and what are the differences? To put the study of genetics into perspective, the pedigrees of animals and plants started 6,000 years ago with initialization of breeding regimes. In fact, the oldest known genetic records are on horses. On the molecular level, there are 4 major theories on inheritance. The first are pangens, which are vapors and fluids. The second is preformation. Ovist animalculist is the third theory, and particulate is the 4th major theory on inheritance. Now, let's explore these theories in more detail, starting with pangens. The first theory of pangens was first proposed by Hippocrates, who lived from 460 to 377 BC. Charles Darwin was notably the biggest supporter of this theory in 1859. In his theory, semen is derived from all parts of the body, and each organ excretes a juice that is transported to gonads. During fertilization, the offspring receives juices from both the female and male parent. In either 1865 or 1866, Gregor Mendel published his work opposing this theory of pangens, but his work was never accepted by Darwin, who supported population control and the theory of pangens until his death in 1882. Another theory, preformation, was proposed by Anaxagoras, who lived from 500 to 428 BC. Anaxagoras was a Greek philosopher of astronomy who discovered the true causes of eclipses, but he was also the first to propose the theory of preformation. In this theory, the embryo is already formed in the female, while the male's role is sex determination. It was thought that supply from the right testicle meant the offspring would be male, while supply from the father's left testicle meant the offspring would be female. A Dutch scientist named Swammerdam supported this theory in 1672, adding that females stored completely formed individuals called homunculi. This was particularly important from a religious perspective, as it meant Eve contained all the people. People of this time had no idea what an embryo was and thought it was a fully formed being. This theory was accepted until the mid-18th century, when Wolff debunked it by showing the fertilized egg of a chicken. Obviously, the egg did not contain a fully formed being. The ovist animalculist theory was first proposed by Democritus, who lived in Greece from 460 to 390 BC. Democritus postulated that the sex of the offspring was determined by the stronger of the two parents at the time of conception. This was opposed by Leonardo da Vinci in 1490, who, based on his observations of interracial marriages, thought both males and females contributed equally. In 1694, Camervis, who studied date palms, claimed that plants had sexes, and opposed the ovist animalculist theory. In 1761, Cole-Reuter demonstrated that both parents did indeed contribute equally to the gender of offspring with his work on tobacco plants. The 4th theory on inheritance, the particulate theory, was proposed by Aristotle, who lived from 384 to 322 BC. Aristotle thought particles were passed from parent to offspring, but he did not actually know what those particles were. He just thought some kind of particle existed that could be transferred. In 1865, Mendel gave the first definite theory for particulates, but seeing how, at that time, the dissemination of information was slow due to the printing press and telegraph being the means of written communication, nobody knew about it, and his work lay dormant until the turn of the 20th century. When it was finally communicated, it led to the following 4 laws: the law of unit characters, the law of dominance, the law of segregation, and the law of independent assortment. The law of unit characters states that contrasting traits of an organism are determined by factors which are passed from parent to offspring via the gametes. Example of this were the contrasting traits of flowers, some being purple, others being white, or some flowers are wrinkly while others are smooth. Those so-called factors are what is now known as genes. Mendel knew about gametes from his work with plants, but he knew nothing about mitosis or DNA, as DNA was only proven to be the genetic material in 1952. Mendel noted that the things passed down were the contrasting traits and that the factors had different forms, now known as alleles. Mendel was also the first to talk about factors existing in pairs. The second law is the law of dominance. Still on the topic of so-called factors with different forms, i.e. alleles, one allele overshadows the other and is said to be dominant, while the one that is overshadowed is recessive. The third law is the law of segregation, and will be explained here in modern terminology. You may have already heard about meiosis, and meiosis is explained in this law as when genes occupying the same locus or position on a homologous pair of chromosomes separate from each other during gamete formation. Mendel had a paired factor, one allele from the female and one allele from the male. He knew it had to separate during the formation of gametes, then come back together from generation to generation. He approached the process mathematically and was ultimately able to solve the issue without any knowledge of cell division and chromosomes. The fourth law is the law of independent assortment. In this law, genes occupying loci, plural for locus, on non-homologous chromosomes separate without respect to each other during meiosis, leading to increasingly more variations throughout generations. Think about the 23 pairs of chromosomes humans have that get scrambled from generation to generation. While Mendel was the first to tie all these laws together, he did not propose these laws. These laws were only formalized much later. Mendel's work stands out because he did not look at whole organisms. He looked at one characteristic at a time, an approach that was much different compared to the other scientists of his time. Mendel did not consider entire flowers. He only considered the flower's color, or the seed color, or only the seed shape. Eventually, these characteristics were looked at together as part of what is known as independent assortment. Mendel worked with true breeding plants, which are plants where all of the offspring appear to be the same as their parent. Mendel inbred these true breeding plants for 2 years before making any crosses. The term paired factors was unheard of before Mendel, but now refers to genes located on chromosomes. In his work on pair factors, Mendel worked with 3 organisms, the garden pea, honeybees, and Oenothera, also known as evening primrose. The Oenothera were messy due to the parent interactions, and honeybees ultimately confused Mendel for the ploidy difference between males and females. You see, male honeybees are haploid, while female honeybees are diploid. Ultimately, the garden pea is the organism that Mendel is the most famous for. Garden peas have 7 traits and 7 pairs of chromosomes. In the garden pea, the traits are far enough apart to act separately. Maybe the traits were not on the same chromosome. Mendel was either lucky or extremely selective because he did not actually know about chromosomes when he studied the garden pea. His work was not immediately accepted by the scientific community, but took off when it was rediscovered in 1900 by 3 scientists independently. Hugo de Vries, a Dutch scientist who worked on Oenothera and coined the term mutations, Karl Correns, a German scientist who studied linkage and genes on the same chromosomes, and Erich von Stramack, a scientist from Austria who studied crosses involving both plants and animals, all cited Mendel's work independently of each other. In 1902, the Englishman William Bateson translated Mendel's paper to English and formally assigned the 4 laws of inheritance to Mendel's work. He described the variations in nature and coined the terms genetics, zygote, homozygote, and heterozygote. He also coined the term allele to mean different forms of the same gene, F1 to mean the first filio generation, and F2 to mean the second filio generation. Other scientists, such as Sutton from the USA, Bovary from Germany, and Weissman, also from Germany, each worked independently but ultimately found the same things. They hypothesized that genes were located on chromosomes and explained meiosis as cell division that produces gametes. They looked at inheritance patterns that corresponded to events happening in meiosis and discovered that fertilization brought separate factors back together. These three scientists are responsible for the chromosome theory of inheritance. Ultimately, meiosis and mitosis were discovered at the same time. In 1910, the second major player in genetics emerged, Thomas Hunt Morgan. He worked with Drosophila melanogaster, more commonly known as fruit flies, and did not believe that genes and chromosomes were related. Ironically, he proved that genes were located on chromosomes. Thomas Hunt Morgan was an early recipient of the Nobel Prize. He also had three students that were quite noteworthy. A. H. Sturtevant was his student that identified the position on chromosomes and mapped chromosome 1. H. J. Muller was Morgan's student that studied the biological effect of radiation and discovered the effect of radiation on genes, a discovery for which he received the Nobel Prize. C. B. Bridges was also a student of Thomas Hunt Morgan. From 1910 to 1930, he worked to map chromosomes 2, 3, and 4 of Drosophila. He was the first to describe crossing over as the way organisms undergo recombination, and was also the first to describe autosomally linked genes, which are genes other than the X or Y chromosomes, as well as sex-linked genes. In the late 1930s to early 1940s, Barbara McClintock described the ACDS system of transposable elements. She called them jumping genes. Most wrongfully, McClintock was not taken seriously by the scientific community. Her ideas of genes moving around on chromosomes or genomes was dismissed, as others said the structure was reasonably fixed. She had her heyday in 1984, though, when she was awarded the Nobel Prize for her work on jumping genes. In the early 1940s, scientists G. W. Beadle and E. L. Tatum used Neurospora, the black fuzzy mold on bread, to put forward the one gene, one enzyme hypothesis, which would later be classified as the one gene, one polypeptide hypothesis. This was the first ever reporting of biochemical genetics, and Beadle and Tatum had figured out what a gene does. In the mid-1980s, transposable elements were found in nearly all living things. This led scientists to finally accept that the genome was not as rigid or defined as previously thought. This acceptance is what led to Barbara McClintock finally being awarded the Nobel Prize. To summarize, up to that point, the major players in genetics were Mendel, Morgan, and McClintock. In the late 1940s, Edward L. Tatum was back with J. Lederberg to study recombination in the bacterium E. coli. They discovered that sex was also a factor for bacteria. In the 1950s, a major breakthrough was made by James D. Watson and Sir Francis H. C. Crick. They discovered the structure and shape of DNA, and DNA was finally accepted to be the source of genetic information in 1952. In 1953, scientists Avery, McLeod, and McCarty, as well as Hershey and Chase, reiterated that DNA is, indeed, the genetic material. The 1960s are when molecular genetics started and was the era that birthed molecular biology and biotechnology. At this time, Crick proposed the central dogma of genetics to explain how the information proposed in DNA is actually expressed. The central dogma of genetics says that the flow of information in the cell is from DNA to RNA to protein. Therefore, RNA contains information for the amino acid sequence of proteins. The DNA replication process was beginning to be examined in the 1960s, but this process is still part of ongoing research today. The genetic code was deciphered in the 60s. It tells how RNA specifies amino acids in proteins. Also learned in the 60s was the 3D shape of tRNA and ribosomes. They were found to play an important role in the conversion of RNA information into proteins. It took 8 years to figure out the structure of the tRNA molecule, and it took 8 to 9 years to break the genetic code. There are 20 amino acids, and researchers had to try to code with all 20 amino acids to figure out the genetic code. In the 1970s, reconstruction endonucleases were discovered. These are enzymes that recognize specific sequences in the genetic code and cut at the same place every time. Also developed in this decade was gene mapping, made possible by restriction endonuclease, which could cut out specific genes. Then came the cloning of genes using the newly developed recombinant DNA technology. The 1970s then saw the meeting of scientists at the Asilomar Conference in California, where NIH guidelines for recombinant research were developed. It was established that these experiments must be followed for cloning experiments. Though originally quite strict, these guidelines were relaxed after a scientist named Roy Curtis III developed a strain of E. coli named X1776 that did not survive inside or outside the lab. The 1970s were all about DNA sequencing, isolating and examining specific genes, and learning about the A, T, C, and Gs of DNA. Then came the 1980s. This decade ushered in the industrial application of biotechnology. Research that had previously only been done in universities and was published exclusively in journals now became proprietary, as it was now being patented by private business. Many ethical issues surrounding biotechnology arose during this period. More positively, the 1980s were responsible for the automation of processes. This includes automated DNA syntheses, automated DNA sequencers, automated pipetting processes, and others. The 1980s also saw the advent of probing, wherein specific genes could be searched for. This is when the genes causing cancer, oncogenes, were first learned about. Scientists discovered that humans have lots of oncogenes, and problems arise when those oncogenes mutate. The beginnings of human gene therapy started in the 1990s. Scientists could identify triplet repeats in the genetic code that could lead to genetic disorders such as myotonic dystrophy. The 90s were the start of the enormous project to map the human chromosome, known as the Human Genome Project. It was very quickly discovered that humans are more complicated than any of the previous test subjects whose genomes were successfully mapped. Most famously to emerge from the 1990s was Dolly, the sheep who was touted as the first successfully cloned animal. However, Dolly, the sheep, was not actually the first animal to be cloned. That title belongs to frogs in the 1960s. Dolly lived until 2003, when she was euthanized for progressive lung disease. The sequencing of the human genome continued into the 2000s, and the mapping or sequencing of biologically involved organisms such as E. coli, C. elegans, and Drosophila began. Stem cell research was also initiated in the first decade of the 21st century, and DNA was starting to be used in forensics. Scientists had established that DNA was genetic material, and it was basically a linear array of genes. Those genes produced proteins as a product, and those proteins then determined what a cell could or could not do. They found that DNA is organized into structures known as chromosomes. Those chromosomes are the vehicle that transmit information between generations and from parent to offspring. Their transmission must be both precise and accurate, otherwise the next generation of offspring would experience many issues. In eukaryotes, the transmission of information involves both mitosis and meiosis. Mitosis is when one singular nuclear division occurs, resulting in the production of two daughter cells that both have the exact same information as the parent. More simply put, one parent splits into two identical daughters. Meiosis is when cell division occurs in specialized cells, resulting in gamete formation with half the number of chromosomes contained in the gametes than what the daughters contained. Whether the transmission was through meiosis or mitosis, both processes see defined and distinct changes occur in the structure and appearance of chromosomes. Now, more about chromosomes. Chromosomes are composed of chromatin, both DNA and protein. They become shorter and fatter during meiosis and mitosis, and the identically individual chromosomes wind up in the nucleus of the cell. Each chromosome has two arms, with one arm being noticeably shorter than the other. The shorter arm is known as the P arm, P for petite. The longer arm is called the Q arm because Q comes after P in the alphabet. As my genetics professor liked to say, mind your P's and Q's of chromosomes. The two arms are joined at one constricted point known as the centromere. Both the position of that centromere and the length of the chromosome's arms are used to classify chromosomes. By convention, the chromosome is depicted by having the P arm above the centromere and the Q arm below. One example of the chromosome classification being used is for metacentric chromosomes. These chromosomes have arms of equal length and a centromere located in the middle. They appear in the metaphase, once DNA has already duplicated. Then comes the anaphase, when sister chromatids will separate to form the chromosome. Also included in the nomenclature of chromosomes is submetacentric. This is when the two arms are unequal and the centromere is closer to one end, between the middle and end. Acrocentric is when the centromere is closer to the end. Acrocentric chromosomes will have extremely short P arms. Telocentric is when the centromere is so near the end that the P arm is not even visible. Only one arm of the sister chromatids, the Q arm, can be seen. Eukaryotic chromosomes show that members of the same species have somatic cells, which contain the same number of chromosomes. The term somatic cells refers to any cell other than gametes. Each somatic cell contains two copies of the chromosome in the nucleus. Therefore, they represent the diploid number, 2n, of chromosomes. Here, n is the number of sets of chromosomes, except for the sex-determining chromosomes X and Y. The diploid number is 2n, while the haploid number is 1n. Haploid is considered to be the genome for the species as it forms the basis of information a species contains. Chromosomes exist in pairs and are called homologous chromosomes. Each chromosome has a specific length and centromere position, and there will be another that is exactly the same. For humans, 2n equals 46 chromosomes. 46 is the diploid number. Then, dividing by 2 gives 23, the haploid number. For comparison, fruit flies have 2n equals 4. The somatic cells of fruit flies have 8 chromosomes. Mitosis is extremely important, and for single-celled organisms, mitosis represents a complete mode of reproduction. Our life begins as a zygote, which is a single cell. Mitosis represents the mode of growth and development for these zygotes. It also represents the method of cellular replacement and womb healing when we are older. Single nuclear division is the division of one nucleus, also known as karyokinesis. It produces two daughter cells, each containing an identical copy of genetic information to that contained in the parent cell. Karyokinesis is then followed by cytokinesis, the division of the cytoplasm. The cell cycle has two phases, the interphase and the separation by mitosis. It is a dynamic process where all stages flow together. The interphase is the phase the cell is in most of the time, and mitosis is when the nucleus divides. The interphase takes hours and has 3 stages, G1, which lasts for 5 hours, S, that lasts for 7 hours, and G2, which takes 3 hours. G1 gets its name for being the first gap. Cells take one of two paths. Either they follow the cycle of staying in G1, going to interphase, then going through mitosis, or they leave the cycle and enter a stage called G0 to become a non-dividing cell. Brain cells are an example of cells that become non-dividing. Some brain cells enter G0 and never re-enter the cell cycle again. They are termed post-mitotic. Others will re-enter the cycle again via G1 after a stimulation such as cancer. G1 also represents the metabolic portion of the cell, as this is where growth and differentiation is very noticeable. Next is the S stage, short for synthesis. This is the only time when DNA replication occurs. Finally, the second gap, or G2, occurs. The cell prepares for mitotic division and will have roughly doubled in size during G2. While interphase can take hours, mitosis takes minutes. It consists of 36 minutes for prophase, where the chromosomes are condensed, then 3 minutes for metaphase, 3 minutes for anaphase, and 18 minutes for telophase. Mitosis can be remembered as duplication division. First, there are 5 stages of karyokinesis, or division of the nucleus. Then cytokinesis occurs, where cells fall into G1 of interphase, and the chromosomes are elongated and perform their normal function. The 5 stages of karyokinesis are prophase, prometaphase, metaphase, anaphase, and telophase. Prophase is an early event in animals. The centriole is split and migrates to opposite ends of the cell, to the nucleus in this case. As the centrioles migrate, the nuclear envelope begins to break down and the nucleolus disappears. The chromatin begins to condense and coil, and actual chromosomes appear in the nuclear area. The chromatin begins to condense and coil, and actual chromosomes appear in the nuclear area. Near the end of prophase, each chromosome is in a double structure when viewed longitudinally, that is. They will have a centromere and sister chromatids, 2 parts that are genetically identical because the DNA is formed by replication that occurred during the S phase of interphase. The sister chromatids are held together by cohesin, a multi-subunit protein complex, at the point of constriction. Prometaphase comes at the end of prophase. Here, fibers often depicted as white lines in diagrams of chromosomes form. These are called spindle fibers, and they are made up of microtubules that run from the centriole to the centromere region of the chromosome. They also run from centriole to centriole to form the axis for chromosomal interaction. Next are the prometaphase and metaphase, which are characterized by the migration of chromosomes to the equatorial plate, also known as the metaphase plate, at the midline of the cell. Centromeres line up on the equatorial plate, perpendicular to the spindle. Prometaphase is the period of time when there is chromosomal movement toward the plate, while metaphase is the moment when centromeres are located on the plate. The spindle fibers bind to the kinetochore, a series of proteins layered around the centromere that serve to pull chromosomes to the pole. Once the kinetochore and spindle are attached by kinetochore fibers, the cohesin that holds the sister chromatids together is degraded by separase everywhere except the centromere, where sugosine blocks the digestion of cohesin. As already mentioned, the spindle fibers of the chromosome are microtubule proteins composed of tubulin. They originate out of the centrosome region, which is the area where the centrioles are. Spindles are the microtubules that are responsible for microbial migration and connection at the kinetochore. The kinetochore microtubule has one end at the kinetochore and the other end at the centrosome. This is species dependent, and there will be one for every chromosome. The total number of kinetochore tubules depends on the number of chromosomes the organism has, the 2n number. Metaphase is completed when each centromere is aligned on the metaphase plate and the chromosomes are extended outward from the plate. Then, anaphase can begin. Anaphase is the shortest stage in mitosis and is when the sister chromatids separate. If they separate, it is called disjunction, but they sometimes don't separate in an event known as non-disjunction. During disjunction, the sister chromatids are pulled to opposite ends of the cell, from plate to centrosome region. Disjunction requires sugosine degradation, which blocks the digestion of cohesin at the centromere. The separase can now fully digest the cohesin at the centromere. The, in quotations, daughter chromosomes are pulled to opposite poles. These so-called daughter chromosomes are what used to be the sister chromatids, but they are now separate. Once the separated sister chromatids reach the poles, telophase begins. Telophase is the reverse of prophase. instead of shrinking, the chromosomes start to expand and enlarge until they become unidentifiable. This signifies the beginning of cytokinesis, which will be different for plants and animals because plants have a cell wall. In animals, cytokinesis is the pinching of what was the equatorial plate. This process can be summarized as cell furrowing, pinching, two daughter cells, interphase, etc. In plants, since they do not have nor do they produce centrioles, a bunch of vesicles appear at the phagoplast, or what was the metaphase plate, instead of cell furrowing. The vesicles release material to form middle lamperi of the cell wall, then open their contents and fuse. They form a cell membrane to create two separate cells, then carry on with interphase and the rest of the cycle. In contrast, meiosis results in the formation of gametes and only occurs in certain specialized cells. Meiosis is a reduction division because it reduces 2N diploid to 1N haploid. It is critical for sexually reproducing organisms. Something that is important to remember about meiosis is that gametes produce half the number of chromosomes compared to the parent cell and compared to somatic cells of that species. Meiosis has two divisions, meiosis I and meiosis II, but both will have the same phases as mitosis, i.e. pro, meta, ana, and telophase. The first division is unique to meiosis and is more complex than the entire process of mitosis. Even though there are two nuclear divisions in meiosis, chromosomal replication only occurs one time, and that is before the first division. Keep in mind that there is no chromosomal replication involved in meiosis II whatsoever. Meiosis I has a few stages. First is the first division, where the homologous chromosomes separate. Then comes the second division, where the sister chromatid separate. Then prophase I begins. The chromatin thickens and coils to produce chromosomes. Then the homologous pairs of chromosomes line up on the equatorial plane and are paired together. The coming together and pairing of homologous chromosomes is called synapse and is where they physically wrap around each other. Each chromosome is bivalent, as we already know, where there are two strands attached to a centromere and two sister chromatids. When chromosomes pair, they become tetrads. Once they become tetrads, crossing over can occur, which allows the exchange of information between homologs. This is eukaryotic recombination. Prophase I has 5 substages. The first is leptonema, or leptotene. This is when the interphase chromatin condenses, so the chromosomes become visible. Bead-like structures called chromomeres will also be visible on the chromosomes. Homologous chromosomes search for each other, like beads on a string. Next comes zygonema, or zygotene. The chromosomes continue to condense, and the homologs continue their initial alignment with each other. Towards the end of this substage, chromosomes are paired with homologs and appear as bivalent. Sister chromatids are not visible yet, though. The third substage is pachynemia, or pachytene. The chromosomes continue to coil and become shorter. Synapsis occurs, and when the homologs are most tightly coiled, crossing over occurs. Each homolog is a double structure, so the sister chromatids of chromosomes of homologous pairs are visible and are tetrads. Crossing over is occurring, and each chromosome will wind up with 2 chromatids. Next is the diplonema, or diplotene substage. Each pair of chromatids begins to separate, but one or more areas of contact remain where the non-sister chromatids have undergone crossing over. These areas of contact are known as chiasmata, or chiasma, when referring to a single area of contact. The 5th and final stage of prophase I is diakinesis. The chromosomes continue to pull apart while the non-sister chromatids remain associated with the chiasmata. As separation continues, association slips more toward the terminal end of the triad to signify terminalization of the chiasmata. Prophase I differs from mitosis in the following ways. First, the sister chromatids remain attached at the centromere in prophase I, and each chromosome's centromere has fiber that attaches to the pole. Also, in prophase 1, each bivalent has its own kinetochore fiber. After prophase 1 in meiosis 1, metaphase 1 proceeds. A chiasmata holds the homologs together, while each triad moves to the equatorial plate. They align at random with respect to which member of the pair lines up to which pole, one chromosome from mom and one from dad. More variation occurs on the basis of crossing over. On average, there is one crossover per arm, per chromosome, per division. This means 46 crossovers occur in every cell in every division. After metaphase 1 is anaphase 1. The cohesin is degraded between sister chromatids everywhere except at the centromere, because the centromere is protected by the Shigoshin complex. This allows the chromosomes to separate. Disjunction occurs, and one half of the tetrad, the dyad, is pulled to the pole. The homologous pairs then separate. Sometimes errors may occur and cause non-disjunction. This is the failure of homologous pairs to separate, because both pairs go to the same pole. The result is one cell getting 2 chromosomes and one not getting any. This can cause many problems. Telophase 1 commences when the chromosomes reach opposite poles and form around dyads. They enter a short interphase that does not include any replication of DNA, because that already occurred, and no S phase. Now for meiosis 2. Meiosis 2 begins with prophase 2 and deals with dyads. Each chromosome consists of 2 sister chromatids that have a common centromere. Their nuclear envelope disintegrates, and the dyads line up on the equatorial plane. In anaphase 2, the chromatids separate because the Shigoshin degrades and the separase does its job. The chromatids are pulled to opposite poles, and the nuclear envelope reforms. One pair of homologous chromosomes contains haploid cells that develop into gametes. Males and females have different reproductive cells that have different processes. Males have cells called spermatogonium cells, which contain one diploid. They undergo growth and maturation that begins with a primary spermatocyte. Meiosis 1 occurs, then 2 secondary spermatocytes are present. Meiosis 2 occurs to produce 4 spermatids, which ultimately form one N spermatozoa. Females do not have spermatogonium cells. Instead, they have oogoniums, which are gonadal cells that ultimately form egg cells. The oogonium is what provides to the zygote after it is fertilized. These cells also undergo growth and maturation. Starting with the primary oocyte, meiosis 1 occurs. Most proceeds go to one secondary oocyte, but the rest go to the first polar body, a haploid. Meiosis 2 then occurs, but this can take 10 to 15 years. Most proceeds go to one cell, but the rest go to a second polar body. Ootids and ovum are involved, and then the first and second polar bodies either dissolve or are removed, but they do not undergo division. To summarize, this episode was an introduction to genetics that covered the history of genetics as a science, beginning with early hypotheses, then arriving at the study of genetics as it is known today. We also discussed meiosis and mitosis and the 2 phases of the cell cycle. Thank you for listening.